Hello, my name is Eliana Vallmitjana and I'm going to talk about bioinformatics pipeline for understanding gene expression and alternative splicing alterations in colorectal cancer. Data we used was downloaded from the Cancer Genome Atlas. Samples are from tumoral and normal tissue. Most of the tumoral samples come from colon adenocarcinoma and the rest are from rectum adenocarcinoma. We analyze expression data with this amount of genes and isoforms respectively. Those genes and isoforms with zero expression values across all the samples were discarded. Tumoral samples were randomly divided to generate 12 independent subsets due to the imbalance between tumoral and normal samples. So we have 12 balanced sub-experiments composed by each tumoral subset of samples and the 49 normal samples. We kept genes with at least 30 CPM per condition and 1,500 reads across all the samples of each sub-experiment and isoforms with an average expression per condition of at least 1 CPM and a minimum relative expression of 0.01 across all the samples of each sub-experiment. General linear models at the gene level were fit using HR. The following criteria was applied to obtain the differential expressed genes. Generalized linear models at the isoform level were fit with NB-splice. The following criteria was applied to obtain the differential spliced genes. The 12 sub-experiments were independently analyzed, and the obtained results were compared to obtain the set of differential expressed genes and differential spliced genes detected in all the sub-experiments. The pipeline proposed by multi-class for identifying samples clusters was used for grouping tumoral samples. 2 clustering were obtained and then contrasted at the consensus step. The first clustering was based on the DEG, and the second one combined both DEG plus the differential spliced isoform that comes from the DSG. Consensus clusters with at least 5% of tumoral samples were kept. 518 tumoral samples were classified into 8 potential colorectal cancer subtypes. DEG and DSG between potential colorectal cancer subtypes and normal samples were identified. Differential expressed genes and differential spliced genes exclusively found in a single subtype normal contrast were identified. Overrepresentation analysis conducted considering DEG and DSG commonly found for the X subtypes identified several processes deregulated in cancer, such as the cyclic GMP protein kinase G cascade, an endogenous apoptotic pathway in colorectal cancer. Functional annotation of exclusive DEG and DSG found for the X subtypes was analyzed. Functional annotation of exclusive DSG of the X subtypes were explored. Here are some examples of represented processes and pathway and the involved genes, most of them that have been previously related to colorectal cancer. Finally, we can say these methods have demonstrated to be useful for discovering and characterizing potential colorectal cancer subtypes.